Hey, Ivy, if you're talking, we can't hear you.
Oh, we were still on mute. Okay.
Yeah.
Now the camera.
Unless you.
Yeah, I'll repeat the question.
Oh, really? Okay.
Oh
this is
yeah,
true.
that's always
actually a good point.
yeah.
We'll have to repeat the questions.
Right. Well, I guess if anyone's in the back and wants to move up to an open seat that is like free, then go ahead. Oh, also, we figured out the image issue. It will not happen again. We figured it out. You guys are good. And just, it's okay. It's going to be okay.
Yeah, I can start. All right. Look at me right there on video. Just terrible. All right, so can everyone in the Teams meeting actually hear?
Yes, you just sound a little low.
Not use that door? Y'all get that?
Go out
Y'all
through
heard
the
that?
last 3 doors, not this one if you're going out because they're working out there, and I guess there's
So everyone on Teams, you don't have to worry about it. Everyone in the actual lecture hall, there's construction going on right there, so use the back doors to exit. Should make sense to everyone, right? Good. All right, so for the people on Teams, sorry if I'm muffled. That is the face mask that you can see. All right, so our very first question, some people did okay on this one, and some people seem to get a little confused. So an infant is consulted by a geneticist at birth. He has a characteristic facial pattern, including low set ears, and he also has a low blood calcium level. The doctor does a chest x-ray and notes no thymus in the film. So right there, that is striking. Everything before that doesn't concern you. You haven't been taught about that. That's not in any lecture. One thing in lecture that's been talked about here is thymus. So there you go, no thymus in the film. Keep that in the back of your mind. He explains to the parents that the baby has an immune deficiency called DeGeorge syndrome. So she did talk about that in the lecture. A decrease in which of the following would be predicted for this child. So if they have no thymus, we're looking at no T cell education. So if we have no T cell education, our biggest thing is going to be we can't kill viruses. That makes sense to everyone? So the alternative complement system here, we know that actually activates without antibodies. So we wouldn't see a problem with that. MAC, the actual MAC complex, that's also part of our innate system. So we wouldn't see a problem with that. And then C3 to C3b, so the activated form, we know that's also not going to cause a problem because that doesn't have anything to do with thymus and T cell education. All right, so our next question, what immune globulin would be used by the immune system to provide the best response in helminth-infected patients? So I know she kind of glossed over it in lecture, and I've talked about it in my review sessions. If you've been to my review sessions multiple times, how important it is to know the antibodies, the immune globulins, and know what their function is. So IgE is allergy and worm infections, so helminth infections. IgA, mucosal. IgG, it's the most abundant. It's kind of the workhorse. And then IgM being the first one made. So make sure you review those and you know exactly which one does which. A patient comes to your clinic after traveling overseas. They express to you their symptoms of stomach pain and diarrhea. Your first thought is a possible helminth infection from undercooked food in the other countries. You decide to do a CBC, a complete blood count with differential blood tests to affirm your theory. What cell would you see elevated in the CBC? So she mentioned this again yesterday when they were talking about immune systems or immune responses to pathogens. So for helminths, we see increased IgE, which then leads to an increase in eosinophil action. Okay, make sure you know which cells are responsible for which battle for our immune system. All right, so this oily compound is found in poison oak, ivy, and mango skin. Your patient explains to you that the following night she had developed a rash around her mouth and on her hands. The last thing she did before bed was eat fresh mango with her bare hands. The following night she had developed a rash around her mouth and on her hands. What is another example of this class of hypersensitivity? So seeing here that it's taken a while, so it's delayed immediately in your head should take you straight to type 4 hypersensitivity. So type 4 are other examples being the TB tests, the tuberculosis test. So Graves, asthma, and systemic lupus, these are different types. Make sure you go through these and you know the examples. I said this in my review. It is very important to know the examples of the types of hypersensitivity. Because if they ask a question to you and they're saying, what are other examples, just like this question, you need to be able to rattle those off. Remember, all of these questions have been vetted through Dr. Pross, so these are very indicative of what you're going to see on your first test. So which antibody is produced first in an immune response? Awesome. Finally got an 80%. So yes, IgM, I can go through these again. IgD, you see that on naive B cells, and it's just stuck to the surface. It doesn't do much. It acts just like a normal receptor. IgG, it's the workhorse, and IgA we find in mucosal surfaces. Which of the following would not be targeted by CD8 cytotoxic T cells? So this is actually pretty similar to a question that I had in my review, Wednesday. Today's Friday. So cytotoxic T cells, when we see that, we should automatically go through that process of where they belong in our flowchart. So cytotoxic T cells, that's going to be endogenous. So it's asking which would not be targeted. So which of these would not be endogenous? So Staph aureus, because it's a bacteria, and I say here extracellular. It's pretty important to note extracellular. That's exogenous, so that's going to be CD4. That's going to be the MHC class 2. Make sure to note that it's not just bacteria, it is extracellular. That's why I put intracellular here for chlamydia. Make sure you also go through that list of the intracellular bacteria that she had in her slides from yesterday. You're going to need to know that because they won't always put intracellular or extracellular. A newborn baby undergoes genetic testing, and the results show he is positive for an extremely rare condition called Bayer lymphocyte syndrome, in which MHC class 2 molecules are unable to be properly expressed. This disease would interfere most with the processing of which of the following types of antigens. So MHC class 2, once again, go through your entire list of what you've learned up to this point. MHC class 2 is what? It's going to be exogenous, and that's also going to lead to CD4 activation, and we're going to create antibodies. So if it's exogenous, the only one here that's exogenous being the extracellular bacteria. An exogenous extracellular protein is being taken into vesicular compartments of an antigen-presenting cell to be broken down. In order for these antigens to bind to the appropriate MHC molecule, which of the following needs to happen? So it's exogenous. Even though it is a protein, it's exogenous extracellular, so that automatically means MHC class 2. Remember, all MHCs are made in the endoplasmic reticulum. We went through that with the chart on using TAP and Tepacin for MHC class 1 and CLIP and invariant chain for MHC class 2. And it's all done in the endoplasmic reticulum. So make sure you note that, and we have those slides. Child is brought into the ER where the doctor suspects she is suffering from a bacterial infection. A polysaccharide encapsulated bacterium is detected through a lung sputum test. Which of the following best describes the immunological response made to the polysaccharide antigen? So we note here it's polysaccharide antigen. That's the main common thing in your question stem. So that should immediately take you to this is T cell independent. We went through that between T cell dependent and independent. So T cell dependent are peptides. T cell independent is anything other than a peptide. So if it's T cell independent, that means T cells and B cells are not going to interact. So we don't get class switching. So we're stuck with IgM. A young female complains of cold sores and fever blisters around her mouth, and you detect HSV1, so herpes simplex 1. What is one method that allows herpes to evade the immune system? This one came straight from her slides yesterday. So what herpes actually does, one of the methods that it actually does to evade the immune system is interferes with TAP. If it interferes with TAP, we know TAP is the connection between Tepacin and MHC class 1. So if it interferes with that, then we won't get MHC class 1 presentation on the cell surface. Antibodies have two basic functions. One part recognizes antigens, the other part is involved in effector function. Which of the following is a correct description of antibody function? So this is just telling you here the different parts. She actually asked this question in lecture yesterday when she asked which part attaches to the microbe and which part attaches to the effector cell. So FAB is going to attach to the microbe. FCR, so the constant region, is going to attach to the effector cell. So the effector cell here we're talking about is going to be the NK cell. She has an actual diagram or a cartoon image, as she likes to call it, in her slides, where we see the FCR attaching to the NK cell, and then that's going to initiate the release of granzymes and porphyrins. Which of the following is not one of the major effector actions of antibodies? So go through this. We know that the whole point of them is for neutralization of toxins, opsonization of microbes, and then antibody-dependent cytotoxicity, so bringing in NK cells to destroy. So they do not inhibit the classical complement system. If anything, they actually activate the classical complement system. All right, does anyone have any other questions regarding my wall questions?
Could you go over the FAB and FCR question again?
Yes. So the question, I couldn't hear it very well. So the FAB is just the region that attaches on to the microbe, and the FC region, that's the constant region, attaches onto the effector cell. So she had an image in her slides where we had a microbe, a bacteria, a virally infected cell, and then she had the antibody actually attaching onto it. It attaches with the FAB region, which is also known as the variable region. And then the bottom part of the antibody, so the bottom stick of the Y, the FC region was kind of sticking out, prepared to attach a NK cell to allow the NK cell to actually go through and induce apoptosis. Any other
Okay, thank you.
I have a question. Can you just explain again what happens when you basically don't get, let me see if I can find which question. I don't think it's related specifically to a question, but basically I remember talking about what happens if the T cells can't do their job. B cells have to step in and bind, and you don't get a really good response.
Okay, so the question from actually inside the lecture hall is to explain the, I'm assuming, the difference between when you have B cells and T cells interacting, or when you have B cells just acting by themselves.
Yeah, so like a T cell independent response.
Okay, so for a T cell independent response, we know that's when you have an antigen that is not a peptide. So if the antigen, if the microbe is expressing polysaccharides or any sort of lipid on its surface, and that's what we break down. When we go through and we present that, that won't activate a T cell response. Remember, a T cell response is necessary to get class switching. So if we don't have a T cell response, if it's a polysaccharide, lipid, nucleic acid, anything like that, and we don't have T cell response, that means the B cells are going to be creating antibodies, but they're only going to create IgM, right? And IgM is not specific enough, and it's not strong enough to actually kill the microbe. IgG is the most effective at actually killing pathogens or microbes, because IgG can, and it's IgG is actually the one that we're going to see in this question that we have on the screen, the one that's actually recruiting NK cells and macrophage. That's IgG's job. So it's the most effective at actually calling, recruiting, working to attract cells. If we can't produce that IgG, then we're stuck with IgM, and IgM does not do a great job of that. Also, and you're going to get into this a little bit later, when you see the pictures of our different antibodies, our different Igs, IgM is the largest one. So IgM being the pentamer, it's the huge one. That can't get everywhere. So it can't travel through the vasculature as easily as IgG or IgA can. So that's going to be another thing that's making that T cell independent reaction not as strong or as effective. Does that make sense?
Yeah, that helps a lot. Thanks.
No problem.
Hey, Daniel, I have one more question about that with the IgG. I was wondering if, so you were saying how IgG is a better antibody. Is that because of the high affinity maturation?
Not necessarily the high affinity maturation. So IgG is better because its actual function. So it's not the affinity maturation can occur in any antibody, and that just makes it have a higher affinity for the microbe and the receptors on the microbe. IgG itself, its actual function is to activate more cells in the immune response.
Okay, thank you.
And then hold on to the teams. I have one question in the actual lecture. So can you say that again? Sorry. Okay, so for the people in teams, the question was revolving around IgM and it being a pentamer and how it's released. And I know that's something that Dr. Prost kind of like glossed over, and it's because they're not really worried about the chemical structure as much. What you do need to know is that IgM is a pentamer, so it's going to be the largest one. And the reason it's a pentamer is because it's the first made. If we have the largest one made at the beginning, then when we're going through class switching or isotype switching, we can kind of pick and choose which pieces of that pentamer we're going to make to turn into IgG, which is a monomer. Does that make sense? Okay. Oh, someone wants number 1 and number 10. I wish we had numbers on these. Okay. All right, so number 1. Okay, so number 1, the biggest thing here in the question stem is that it says no thymus. So if you have no thymus, we should know that thymus is the portion in the body that does primary education for our T cells. And we know that T cells are the biggest proponents in actually fighting intracellular pathogens, whether it be a virus or intracellular bacteria or cancer. So if we have no education in the thymus, that means we're going to have no education for T cells, which means we will lack that ability to actively kill viruses. So we're going to lose that CD8 education. The alternative complement system is not, it's not enacted by any sort of T cell or B cell. The alternative is just the innate, so it's just kind of free roaming around zymogen components that are going to break down and activate the alternative complement system, so it has nothing to do with the thymus. The MAC, membrane attack complex, also has nothing to do with the thymus. It's part of our innate, and then C3 and C3b is also part of our complement system, so it doesn't have any direct action with the thymus. Okay, and then someone wants me to go over number 10 and number 9. So I'm going to go over number 9 first. Okay, I think I did that right. Oh, well, luckily we kind of already went through this. So for number 9, IgM and T lymphocyte independent. We know it's T lymphocyte independent. It's T cell independent because it's a polysaccharide antigen. So peptide antigens would give us T cell dependent. And if we have T cell independent, that means the T cell and the B cell are not working together, which means we do not get class switching, which leaves us with IgM, the first one made. Okay. We'll go to question 10. A young female complains of cold sores, fever, blisters. So this one came directly from the slide of what herpes does to avoid or to evade the immune system. So make sure you go through that. It's not anything I can really review other than know that information from the slide. This last lecture is probably going to be the most important for the upcoming test, this lecture, and then as well as Tuesdays for Dr. Pross. After that, you'll have questions from Dr. Anderson. Those will be a little bit more straightforward. Okay. We're going nuts here.
One quick question, if you don't mind me asking. For number 6, tumor antigen is on that slide of one of the things that CD8 like attaches to and kills, because it would be like a tissue cell that's presenting, hey, I'm infected, so kill me, right?
Right. So the question's asking which one would not be targeted by CD8. So CD8 cells do target
I missed that. Thank you.
viruses or tumors or any intracellular bacteria. The question's asking which one does not.
I actually have a question as well, referring to the FC region one again. In the PowerPoint, FCR was listed as the receptor on effector cells, but here it's like in the question it was said to be part of the antibody. So I just wanted to. That was question 11.
Okay, so right here.
Yeah. So it said then the PowerPoint that FCR was the receptor on that looks for FC on the effector cell, and that FC was the portion of the antibody. So I just wanted to clarify that.
Okay, yeah. So for clarification on the actual image and the antibody itself, the antibody has the FAB and the FC region. The FCR is the receptor region for the FC, which is on the effector cell. Yes.
Okay, thank you very much.
No problem. And that is pretty much explained. All right. Does anyone else have any questions for me here?
I have one more. It kind of wraps back around to my last question.
Sure.
So like when you're not getting a T cell response, I know she used the example with Streptococcus pyogenes with the M protein.
Mhm.
So when you're getting an IgM, basically, since you don't have T cell activation, is it because of that lack of specificity you can go on to get like rheumatic fever and such?
No, rheumatic fever was something different. That comes from molecular mimicry. So for the people in teams, the question was revolving around the T cell independent response and how that might lead with bacterial infections, the actual strep example that Dr. Pross gave in lecture for rheumatic fever. So rheumatic fever is completely separate. So that's creating antibodies that cross-react. So what we
She
have
mentioned it
is,
on the same slide yesterday.
what happened?
She mentioned them both on the same slide yesterday.
For which one?
For the M protein, she mentioned strep and rheumatic.
Right. So yeah, so the M protein specifically is inhibiting something in our immune system. So by inhibiting that activation, I think it's C3, by stopping that C3, it's allowing itself, it's an evolutionary process. So that way the strep can survive longer. As it survives longer, that's going to lead into rheumatic fever because we're creating that antibody to the strep that's cross-reacting against the cardiac myocytes.
Yeah, that was my question.
Did I answer it?
Yeah, you said I was wrong, but now you just said
Well, I
what I was
didn't-
asking you. How you basically you could get rheumatic fever from that,
From,
from strep.
yeah, I didn't say that you couldn't get it. I was saying,
You just said they were two completely different things, so I assumed that I was wrong.
okay, sorry that you assumed that you were
Oh,
wrong.
it's all good.
I said it in my review. If you guys were there on Wednesday, I did say that. So the whole point when she brought up Little Women and wanted to talk about that, she was talking about how it extends to one, and that's the example of molecular mimicry. But yeah. Does anyone else have any other questions?
Can you please explain why the inhibition of the
one second.
classical?
Sorry.
Can you please?
Give me 1 second. I have one in the actual lecture hall. The last lecture. So the one that she did on Thursday, she has a slide where she talks about intracellular bacteria and she just kind of lists them off. I think it's like 5 of them that she just straight lists. No problem. All right, sorry. Go ahead, Lordina. What was your question?
I was asking if you could please explain why the inhibition of the classical system is not a major effect of the antibodies again.
For which question? If you don't mind me asking.
It was about the ones that are not an effect of the antibodies. I forget what number. Yeah, the
Okay,
question right
so,
here. Yeah.
This question. So which of the following is not a major effect of action of antibodies? So inhibition of the classical complement, because the classical complement system is activated by antibodies.
Oh, okay. Thank you.
No problem. Any other questions? All right, I'm going to pass it off to our next EA. Phoebe.
Hey guys, nice to see some of you in person. So most of my questions were pretty just memorization questions. So if you didn't do too great, just it's no big deal because it's not really conceptual. But anyways, we'll start with the first question. Which of the following is false regarding de novo synthesis of a purine? So the commitment step is catalyzed by PRPP synthetase. She said that the commitment step is actually the glutamine aminotransferase or whatever. So that would be wrong. So that's why that is the correct answer. It occurs in the cytoplasm of hepatic cells. We know that the de novo synthesis of purines happens in the liver, specifically in the cytoplasm. The commitment step requires PRPP and glutamine. Yes, these are the two reactants that are used to do the commitment step. So that's true. And the formation of AMP requires GTP. So yes, remember that AMP needs GTP and ATP needs GMP. No, GMP needs ATP. So it's the opposite. So you guys did pretty good on that one. Okay, so the next question. A researcher is trying to synthetically create GMP. What is the proper order in which he needs to add the enzymes for each step? So this is just pretty much you reiterating what the order of the enzymes are to make GMP. So you've got your first step that you have to know. This is the enzyme for it is PRS. Remember that it makes PRPP. The second step, which is the commitment step, and this is the glutamine PRPP aminotransferase. And so you know that's the second step. So that has to be correct. So you can cancel out the last two options. Then you've got these two arrows indicating those multiple steps that you don't have to memorize. And then finally, you have IMP as the product after those multiple steps. So from there, you have to determine which pathway between the GMP and the AMP pathways you need to take in order to get GMP. So you know that the IMP dehydrogenase is the enzyme that's going to ultimately give you GMP. The adenylosuccinate synthetase, however, is going to give you AMP. So you just have to differentiate between those two. But it looks like a lot of you got that right. So you guys are good on that. The third question is the committed step for the pyrimidine and purine de novo pathways involve which of these enzymes respectively. So this is another memorization question. It's really just telling you which is the enzyme for the committed step for pyrimidine, which we know is the ACTase. And then you have to know the committed step enzyme for purines, which is glutamine PRPP aminotransferase. So really just a memorization question. Your PRS is going to be your first step, so that's wrong. Your CPS is also going to be your first step for the, I think, pyrimidine synthesis, so that's also wrong. But it looks like most of you got this right, so that's good. The next question, the final two steps for pyrimidine de novo synthesis involves a bifunctional enzyme with a functional N-terminus and C-terminus. What is the product of each of these enzymes? So for this, you're really just having to remember the last two steps to make UMP. So you know that the ultimate product is going to be UMP, hence why the C-terminus yields UMP. So that has to be the right answer, or that you can cross off the last two because they don't yield OMP as a final product. So you also have to know what the second to last step to make UMP is. And from there, you just have to understand that the N-terminus is the one that does the second to last step, and the C-terminus does the last step. So, and understanding that OMP is the second to last product before you get UMP. A few of you got it a little bit backwards. So orotic acid would be the third to last step. So it goes orotic acid, followed by OMP, followed by UMP. So yeah, you just switched it, but that's okay. All right, this question is characterized by megaloblastic anemia, orotic crystalluria, and nephropathy due to accumulation of orotic acid. This condition is treated by, so you have to know that this is going to be the dysfunction of the bifunctional enzyme that we talked about, which is the uridine 5 monophosphate synthase. And it causes the accumulation of orotic acid. So that's your key word here. And this is the orotic acid urea, or hereditary orotic acid urea. So it's the pyrimidine synthesis metabolic disorder that you have to memorize. We know that allopurinol and the NSAIDs and all these anti-inflammatory drugs are associated with gout, so that's wrong. And then also probenecid is also associated with gout as well, so you can cross those off, leading Xeridin to be the correct answer. So this is just another memorization question that you have to know. Okay. And the last question I see was a little bit tricky, but that's okay. So on one of the slides, you pretty much get to learn the conversion of a ribonucleotide to a deoxyribonucleotide. So remember that this is going to use the ribonucleotide reductase working with the redox enzymes, GTX or TRX or whatever. So remember that these redox enzymes are going to take the hydrogen groups from the NADPH and break their disulfide bridges. And then once they break those disulfide bridges, they will form disulfide bridges again by handing those hydrogens off to the ribonucleotide reductase so that it can convert the alcohol group on the ribose sugar into a deoxyribose sugar. So yeah, the formation of disulfide bridges is therefore important for that. So it's I guess a little bit of a detail, but it might be important to know. I think that's all of my questions. So does anyone have any questions about that?
Hi, Phoebe. I have a question about your last question.
Yeah.
I think I got tripped up because, for instance, that I know that that reaction has cofactors. However, I was under the assumption that the level of chemistry of having to know its like formation and degradation of disulfide bonds was a little granular. Are they really going to ask that level of chemistry on this biochem exam?
Well, I would say to kind of have an idea as to what's going on this slide, knowing more details than not enough details is better. But if I remember correctly, this test for us, since it was like the last lecture of our block, she kind of gave us easier questions for it, but I'm not sure if she's going to do the same for you guys or if they even use the same questions. So I kind of wanted you ask that in order for you guys to understand it just in case. Does that answer your question?
It does and it doesn't. So basically, Dr. Patel is writing the questions for that lecture, and we'll have to ask her if she's going to that level of granularity with chemistry.
You could if you want to. I mean, like I said, we learned about disulfide bonds before, and she does have it on her slide, essentially, and I'm pretty sure she mentioned it. So it's better to know than to not to know.
I understand, but she mentioned cofactors. She didn't point it out that that was like a major.
The cofactors, I think, are magnesium in this case.
I meant the cofactors as in the TRX and GRX, the actual things that are being reduced, like the disulfide bonds that are being formed and then reduced.
Yeah, those are the enzymes that are being that have the disulfide bonds that are being reduced. So those are just enzymes that work with the ribonucleotide reductase.
Okay, I will email her and then post it in the discussion what she says. Thank you so much.
Yeah, sure. No problem. So somebody asked a question in the chat, is orotic crystalluria not a symptom of gout too? No, remember that gout is uric acid, not orotic acid. So that's the difference. Any other questions? No? Okay, then I'll pass it off to the next TA. Ivy, right?
All right. Hey guys. It's nice to see some of you in person finally. Hopefully I get to see all of you at least at some point. So if you didn't hear me earlier, we fixed the image issue. Shouldn't happen again. If that does happen to you on an exam, just email the course director, and honestly, they're really good about it. If it didn't show up for some people, they usually just like toss the question or like give everybody points because they can't control like who sees it and who doesn't. So it's going to be okay. The first question was, a patient comes into the lab and wants to have their genome sequenced. Upon sequencing, you learn that they have a V600E mutation in the BRAF protein. Knowing what you know from your MSP3 genetics course, what would a V600E mutation cause? Looks like 53% of you put the right answer. So yes, if we have a V600E mutation, once you guys get access to like see the correct answers, I also put my explanations with the slide. I don't know if you guys can already see that. We don't see the student view. Yeah, awesome. So we know that if we have a valine getting switched out for glutamate, glutamate has a negative charge like phosphate groups. And the BRAF protein is basically like turned on when it's phosphorylated. So it's like ready to go, ready to stimulate cellular respiration. So if we have a glutamate in place there with that negative charge, it's like tricking that protein to thinking that we're turned on, we're ready to go, we're dividing. So we're just going to get continuous cellular proliferation. Okay. So the next question. No upper or lower size limit is a characteristic of what type of DNA sequence? So that was on slide 15. A lot of you got that right too. So that's the single copy. That's just listed there with all the characteristics on slide 15. You just kind of have to know those differentiating factors between the single copy, moderately repetitive, and highly repetitive. So once we finish that like PowerPoint, next week, then you'll kind of go into like what the other two are. Which of the following genes is a more likely target for mutation? A lot of you got this right, so that's good. So we know that the larger the gene segment is, the more likely we are to have a random mutation at that gene segment. So that was on slide 16 when he talked about that. So we know that the SRY gene and the hemoglobin gene are relatively small genes because they're important. So if they're smaller, they have less likely of a chance to get a random mutation. And he wanted you guys to memorize those numbers. Like, you guys should memorize those numbers. Please memorize those numbers. He said that's like what he wanted you to know. So please memorize those numbers. Okay. True or false? Gene families cannot be on different chromosomes. That's false. So most of you guys got that right too. They can either be next to each other or they can be dispersed on different chromosomes, and that was listed on slide 17. Most human genes are in which category? So most are in the single copy. That was again also on slide 15 when he listed all the characteristics. You guys should know the characteristics and what differentiates like single copy from moderately repetitive and highly repetitive. And then what is used to define a gene space? So that's what he talked about in the beginning of the lecture, the primary transcript. That was on slide 9. My last question. Yeah, that's my last question. So does anyone else have any questions for me?
I have 2.
You have 2. Okay.
I have 2. I know I'm needy. For 21, in class when you're
Which one is 21?
Memorizing the memorizing base pairs and such. He was like bouncing back and forth between like kilobases and bases. Do you know which one he's going to use? Because he was going from like he was saying 1,000,000, which I think he was just converting from kilos. Do you know which one he's going to use on the exam?
Then it was the one all the way on the left.
But like what units?
What units was the number all the way?
It was in kilobases.
Then kilobases.
Okay, because he was saying it differently after he showed us the slide in
Yeah.
class. So,
Yeah, whatever is on the slide, I have it highlighted for you guys when I publish my notes, that number. If it's kilobases, if that's the units, I can't remember off the top of my head right now, but that's the number that you should know. And you should also just be aware which genes are smaller, which genes are bigger, and what that has like an effect on if the gene can get mutated or not.
Okay.
Yeah.
Cool. Then my other question was in regard to gene families being on different genes. In the pictures and in the slides, it kind of looks like are we talking about different like loci, or are we talking like completely separate genes?
No, they can also be on different chromosomes. I think I wrote exactly what was, or like really close. Yeah, gene families may be clustered together or dispersed on completely different chromosomes. Okay. So just depending on how they evolve, they can evolve right next to each other, or they can evolve to be on different chromosomes.
Okay, cool, thank you.
Uh-huh. The which one? Oh, for this question? The true or false question? Oh, the one that the DMD is the correct answer? That is going to be slide 16. Let me check the chat. Yeah. So I think that will change once we release that you can see like all the answers. So like after this session, we release it so that you can see all the choices. And then once you see all the choices, I'm pretty sure that will show up. I will double check. I can't see it from your view, so I have to kind of ask Rose, or I don't know, but I'll figure that out by the end of the day. Yes. Yes, so let me pull that back up. Okay. What? Oh, it's on my computer. Yeah, I'm on my computer. It's that I can't see my explanation right now. Yeah. So the BRAF protein gets phosphorylated when it's time for proliferation, okay? So phosphate has negative charge, right? So if we're switching out our valine and we're putting a glutamate there, glutamate has a negative charge. So it's basically like tricking us into thinking, oh, I have this negative charge, I'm activated, I want to start proliferating. So then it just keeps proliferating because that's a mutation that's going to stay there. It's not like getting phosphorylated or dephosphorylated. Yeah. Okay, I think I had a question in the chat. Would tumor suppressor genes like P53 be an example of a gene family or a super gene family? I'm not sure. I'm not sure. I don't know how they evolve. Do any of you guys have insight on that? Tumor suppressor genes like P53, are they a gene family or a super gene family? I don't know.
I don't think they qualify.
Yeah. So we don't think they qualify under either. Email Dr. Blank. He would love that question. But that's like beyond the scope of what we need to know. If he's going to pull an example and he's going to ask you to like differentiate, it's probably going to be an example from straight from the PowerPoint. He's pretty straightforward. Okay. No other questions? You're welcome. Okay, if there's no other question, oh, you do have another question. Dr. Blank, so the question was for my people on Teams, the question was if he wants us to know that V600E mutation example, even though it wasn't explicitly written on his slides. You're going to come to find out he doesn't write anything on his slides, and you have to just listen to what he says. So when I was in your shoes, I literally just watched the lecture. If I had to watch it on 0.75 speed so I could write down everything that he said and then filter through it after, that's what I did. So I'm trying to like kind of give you guys like the main idea of whatever he talks about on his slides. And when you guys are like going back and like taking your notes, rewatching the lecture, you can kind of like listen for those main ideas and get what he wants you to know, because he kind of rambles and he doesn't have a lot written on his slide. So you have to kind of try to listen to what he wants you to know. All right. Okay, if there's no other questions, I'll give it to Justin.
All right. So I have the pleasure of giving you guys the last 12 questions. So feel free to just raise your hand or let me know if you have any questions regarding these. So let's see what this is. All right. Okay. So the first question. A patient comes into the clinic and states that they are feeling numbness, tingling, and weakness in their hand. As the physician, you perform several tests and diagnose the patient with carpal tunnel syndrome. So right there, if you know that carpal tunnel syndrome involves the median nerve, you're already halfway through this question. So which muscle is not affected by the injury, right? So if you were at my morning session, you would remember that median, 1/2 loaf. So if you remember median 1/2 loaf, then you'll be able to remember that the fourth lumbrical is not going to be supplied by the median nerve, because 1/2 is going to stand for the first lumbrical, the second lumbrical. The off, which is going to be opponens pollicis, abductor pollicis brevis, and the flexor would be flexor pollicis brevis, right? But only on the thumb side. Remember, so the pinky side is going to be ulnar nerve. This is going to be median nerve. So only the thenar eminence is going to be median nerve, as well as the first lumbrical and the second lumbrical, right? So if we knew that, and if we remember the mnemonic, then we'll be able to tell that the fourth lumbrical is not going to be supplied. All right. Damn, it's so hard. Okay, so for the next one, so what is the muscle and the action of the structure being pointed at by the arrow in the image? So
if we see this image and we see that it's going to be the back of the hand, then we realize that these are going to be the dorsal interosseous muscles, right? So these are going to be your dabs, right? So they're going to allow you to abduct, so allow you to spread your fingers away. These are going to be your dabs, and if you remember, dabs, abduction, so dorsal interosseous, abduction, so B would be the correct answer. All right. So you're in an emergency clinic and a patient is brought in from a severe car accident. As you're checking their symptoms, you notice that they have a wrist strap on their right hand. You take several x-rays and determine that the patient suffered a Holstein-Lewis fracture, which is a fracture of the distal third of the humerus, resulting in entrapment of the nerve that supplies the anconeus muscle. So the whole thing, this entire paragraph, you don't need to worry about what it means. The only thing that you need to know, basically, is what supplies the anconeus muscle, right? So if you know that, you'll be able to answer the question, right? So what supplies which other muscle is most likely affected by this injury? So which nerve supplies the anconeus muscle? Does anybody remember here? Anconeus muscle. Anybody can say it.
Radial nerve.
It's on the posterior compartment of your arm.
Radial.
Yep, radial nerve. Yep. So if you know that it's radial nerve, then you'll be able to figure out that which one of these is going to be innervated by radial nerve will be the one that's going to be affected, which is extensor carpi radialis. Right? So I put flexor carpi radialis just to trick you, because flexor carpi radialis is still ulnar nerve, right? I'm sorry, no, it's still going to be median nerve. And then you have pronator teres, and then abductor pollicis brevis, which won't fit into that category as well. So the answer would be B, extensor carpi radialis. All right. So the muscles in the hypothenar eminence are innervated by which nerve? Okay, so hypothenar. Remember, hypothenar is going to be on the pinky side, right? Because thenar eminence, think T for thumb. Thenar eminence is going to be on the thumb side. So we're talking about the hypothenar, so that has to be on the pinky side. So which nerve is going to be innervated by the hypothenar eminence? It's going to be ulnar, right? So it's only going to be ulnar. So it's going to be 1. That's because if we remember our 1/2 loaf or median 1/2 loaf, then we'll remember the first lumbrical, the second lumbrical, as well as off on the thenar eminence is going to be median nerve, right? The rest of your muscles on your hand are going to be ulnar nerve. So I can say any muscle in the hand, and if it's not 1/2 loaf, then it's going to be ulnar nerve, right? So again, remember I put hypothenar just to make you guys realize that just because I'm saying oaf, I'm talking about the oaf on the thenar eminence that's innervated by median nerve, not the oaf on the hypothenar. That's ulnar nerve, right? So yes, that's going to be A. Any questions on that one? No? Okay. So next one. A parent is arguing with their child at a local Publix and tugs the arm of the child in an attempt to leave the establishment. The child begins to cry and later tells the parent that their elbow is in a lot of pain. The parent takes the child to the physician, and the physician stated that the child has nursemaid's elbow. This is a very common injury in young children that is caused by the dislocation of a bone from the annular ligament. Right there, last line. So remember, if we talked about, I talked about in my lecture, annular ligament is going to surround which bone? Does anybody remember? Radius, right? So that we know that the annular ligament surrounds the radius, and my question is saying which bone is dislocated. So the answer is going to be A, radius. So radius, like we said, when they pull, like they tug the arm of children, usually when they dislocate their elbow, it's going to be their radius popping out of their annular ligament. So yes, that would be correct, A. Okay, so which of the carpal bones is the most distal and lateral? So this is memorization, but I tried to give you guys a mnemonic in the first, or in my session in the morning if you saw. So the mnemonic is going to be some lovers try positions that they can't handle. So you got to do the first 4 in their own category and the last 4 in their own. So what I mean by that is some lovers try positions. So the first 4, if you start from your thumb, you go some lovers try positions. Those are the first 4, which are going to be scaphoid, lunate, some lovers try. That's going to be tirikitarao or tirikitam, whichever way you want to pronounce that, and then pisiform. Those are going to be the first 4, right? Coming from the thumb all the way to the pinky. Then you go superior or distal to that. Let me just say distal. Some lovers try positions, and then you go that they can't handle. So you go back to the thumb up here, that they can't handle, right? So I think everybody can see that, right? Which is going to be the rest, which would be trapezium, trapezoid, capitate, and then hammock. So those would be the last carpal muscle bones. So if you remember those, then you'll be remember that the trapezium is going to be the distal and lateral side. So distal, further away, and lateral because it's on your thumb side. Anything that's on your thumb side is going to be lateral, right? So anything on your pinky side is going to be medial. Regardless if I pronate or supine my hand, it's always going to be lateral on the thumb side and then medial on the pinky side. Any questions on that one? Okay, cool. So all of these are innervated by the radial nerve except, so if you remember, brachioradialis is technically part of the posterior compartment. So brachioradialis is going to be innervated by radial nerve. Anconius, one of our questions already asked about that, posterior compartment, radial nerve. Last one, pronator teres, that's one of our pass-fail. That's going to be on your anterior compartment, and that's going to be median nerve, right? So that's going to be the only one that is not innervated by radial nerve. Any questions on that one? Okay, cool. So you're a college football player running down the field to tackle a play on the opposing team in a championship game. When you finally catch up to the player, you reach out with your hand and tug his jersey so that you can take him to the ground. After that quick tug, you experience some excruciating pain in one of your fingers. The medical team on the field noticed that you're unable to bend the fingertip of your fourth phalange, and they claim that you may have jersey finger. Which muscle is affected due to this injury? So this is actually a pretty common injury that can happen, and it's also most likely going to happen in the fourth phalange. So when I said fingertip, that should be like coming into your head like, bam, that's got to be something I know. Fingertip is going to be the distal end of your phalange. So these will be your fingertips, right? So if you can't flex your fingertip, then you know this has to do with your flexor digitorum profundus, right? So over here, oh yeah, and then that's the answer, right? Flexor digitorum profundus, because they can't flex the distal part of their phalange 4. If I couldn't flex the middle part, which muscle would it be?
Superficialis.
Yes, flexor digitorum superficialis. So if I couldn't flex the middle one, it would be flexor digitorum superficialis. If I couldn't extend my index finger, it would be extensor indicis. And then flexor carpi ulnaris, it would be somewhat associated with flexing, not being able to flex your wrist, as well as deviate it towards the ulnar side. Right, so good job with that. Okay, so which of these muscles has an attachment point to the coronoid process of the ulna? So I tried to make this like a recap question because I wanted you to remember that the pronator teres is not the only muscle that attaches to the coronoid process of the ulna. It's also the brachialis. So I use this question for like study purposes as well to remind you that coronoid process has the pronator teres as well as the brachialis, right? So, and then flexor carpi radialis, that's just going to be the medial epicondyle of the humerus. Does anybody remember the muscles that attach to the coracoid process? Coracoid process of the scapula. Just name one. You don't have to name them all.
Teres minor.
Coracobrachialis. Pec minor.
Small head of the bicep.
Short
The
head
biceps.
biceps.
Yep, and the short head of the biceps brachii. So I would like list those into their own categories just for you to remember. We talked about 3 muscles that attach to the coracoid process of the scapula. That'll be short head of the biceps brachii, the pectoralis minor, as well as the coracobrachialis. So bam, that's one category. The next category is coronoid process of the ulna. That's going to be brachialis as well as pronator teres. So boom, that's its own category. And now you know all the muscles that attach to each. So you can't trick you on the exam which one attaches the coracoid, which one attaches the coronoid, because now you know. All right, cool. So you're running outside and playing tag with your friends, and when one of your friends accidentally pushes you too hard and you fall into the floor with an outstretched arm, you immediately have pain in your hand/wrist area and get it checked out. The clinic diagnoses that you have a navicular bone fracture of the hand. This is a fracture in one of the bones that is directly attached to your radius. Which bone is most likely affected? So the radius is on which side, the thumb or the pinky side? Thumb, right? Okay, so it's got to be one of the bones that's going to be towards the thumb side, right? So if we remember, the sum lovers try positions that they can't handle. So sum is going to be scaphoid. Is that on here? Yep, that's it. That's going to be the answer because that's the one that's closest to the radius, right? The next one would be lunate. I didn't put lunate because lunate's also attached to the radius because then there would be 2 right answers, right? Hamate, trapezium, and trapezoid will be on the distal part, attaching to the metacarpals, right? So those would be the first 4 in that some lovers try positions that they can't handle will be attached to the radius or ulna, while the other 4 will be attached to the metacarpals, right? So that would be scaphoid. Any questions on that one? No? Okay. What is the main function of the palmar interosseous muscles? So when you see this, you got to think pads, right? palmar interosseous muscles, pads. If I said dorsal, it would be DABs, right? So palmar interosseous muscles, pads, then we already know our answer. It would be adduction, right? So abduction will be our dorsal interossea, right? Cool. Nice. So the tendon of which muscle does not go under the flexor retinaculum, right? So which one of these does not go under the flexor retinaculum? We talked about most of these, the flexor carpi radialis, the flexor pollicis longus, and the flexor digitorum profundus all go under the flexor retinaculum. The palmaris longus only goes to what location? Do you guys remember? The distal attachment of the palmaris longus. Yep, the palmar aponeurosis, right? So it's not going to go under your flexor retinaculum. So that's why the palmaris longus will be the only tendon that is listed here that doesn't go through the flexor retinaculum. Yep. That's it. Yeah. Does anybody have any questions?
Justin, for the one that
for
was
27, the question with the wrist drop.
Yeah.
I also thought of it like if you have wrist drop, you're pretty much just doing like that, just like you're doing flexion, right?
Yep.
So you're just doing an antagonistic of extension because you don't have the nerve innervating extensor, carpi radialis. So I just want to make sure that thought process was.
That
right.
thought process is good too. Yeah, because with wrist drop, it's associated with no extension. So that would work.
Okay, thank you.
Yeah.
Just wait, did he? I don't know if he said
Anybody else?
my question was, but I was saying that for that.
Oh yeah.
For that
You
one.
repeat the question again? I didn't hear you.
So the question with the same thing, the pronator teres was what I answered. Was that what he was saying?
So the question was, is nursemaid's elbow and like jersey finger, those things that I wrote, important to know? No, they're just like cool facts for you to understand like the relationships like in real life, like what these things actually are. So for example, jersey finger is common in football, and it's really related to the flexor digitorum profundus, right? So I wanted to like make questions that are kind of like real life so that you really understand like how these muscles work and how injuries are going to affect them. So no, he's not going to test you about jersey finger. If he tests you about anything that's like outside of the scope, there's going to be something in the question stem that leads you to something that you know, right? So for example, like I said that whole thing, but then I was like, okay, then we're talking about the fourth finger, right? So it's not flexing. That's the most important part, right? That's all you need to know to answer the question. The other stuff is just for you to, like, for learning purposes, right?
Justin, can you go over 28 again, please?
Which question is that?
The hypothenar eminence.
Yeah, the hypothenar?
Yeah.
Okay. So if we remember the thenar eminence, okay, so the phrase that you want to remember is median 1/2 loaf, right? So 1/2 stands for the first and second lumbrical, right? And then the oaf is going to stand for opponens pollicis, abductor pollicis brevis, and flexor pollicis brevis. Those are all going to be on the thumb side. Those are going to be innervated by median nerve. So if we know that, any other hand muscle I say is going to be ulnar nerve. Even the hypothenar eminence, that's still ulnar nerve, right? So when I say median 1/2 loaf, that loaf is only talking about the thenar eminence, right? So if we know that, then we see what muscles are going to be innervated by the hypothenar eminence, then we know it's only going to be ulnar nerve. Does that help? Okay.
Yes, thank you.
Yep. So what were the coronoid processes, two groupings again? Okay. So the coronoid process of the ulnar. Can anybody tell me what attaches there? Coronoid process of the ulnar. Yes. So the pronator teres and the brachialis are going to attach to the coronoid process of the ulnar. Yep. And then coracoid process of the scapula is going to be coracobrachialis, short head of the biceps brachii, and pectoralis minor. Any other questions?
Hello? Can you hear me? Hello?
Oh, I can't hear you.
You can't hear me? Oh, man. It might be my speaker. I'm just going to type up the question.
Fernando, we can't hear you.
I'm not tech savvy.
Daniel said he's going to type out his question.
Oh, okay.
While he's doing that, I had a question, Justin.
Okay.
For the running outside and playing tag, with the, could you explain again why it was scaphoid?
Yeah. Hold on. This one. So, yeah, you're running outside, you get perched, you land on your outstretched arm. The most important thing that I put on this question is that the fracture is one of the carpal bones that is directly attached to the radius, right? So you have to know that the carpal bones that attach to the radius. So remember I said if you break up the mnemonic into 4 and 4, sum levers try, then you know the sum levers is going to attach to the radius, right? Because we're starting from the thumb side, right? So from the thumb side, so sum levers, those two are going to attach to the radius. That's going to be scaphoid and lunate. So if I put scaphoid or lunate, that's what was going to be the answer. If I put, some levers try. If I put triquitrao or pisiform, that's going to be associated with the ulna, right? I know, that's the way I pronounce it, whatever. Triquetrum, triquitrao, whatever you guys want to call it. My Hispanic's coming out, okay.
Thanks, man. Appreciate it.
Yeah, I got you. Okay, so if the palmaris longus did go through the flexor retinaculum, we wouldn't be able to tell if we had one, right? You still would be able to tell if you had one. You probably still would be able to tell if you have one because your palmaris longus goes down your midline. So you'll be able to tell if you don't have a tendon going down the midline of your hand, regardless, because you can still see other tendons on your hand other than your palmaris longus. If you just flex hard enough, you'll be able to see other ones. So just because it's going under the flexor retinaculum doesn't mean you won't be able to see it. Okay. Okay, so the anconeus doesn't assist in supination. The reason why is because the anconeus is attached from your humerus only to your ulna. Your ulna doesn't move, right? So since your ulna doesn't move, your anconeus can't assist in supination, right? So since your anconeus can't assist in supination, then it's only going to be an extensor. And since it's an extensor, the only nerve that's going to innervate the extensor is which one? Radial. Right? So radial nerve. So that means that's how we can figure out that radial nerve is going to be the one that's severed for the question that we talked about. Gotcha. Okay. Any other questions?
Does anyone have any questions like regarding anything other than this Q&A session? Study tips? Yeah. Well, well, these, I mean, some people take them seriously, some people don't. So like, if we look at the average of this one, I think it was like in the 50s. Yikes. Yeah, like some people don't even answer, so they get a 0, so it skews. Yeah. But I mean, like in my study group, some of us would get score well, and some of us would get like also in the 50s. We all still came out with A's. It just, if you're like a little bit behind, then you just got to catch up on the weekend. Yes. Oh, I saw her first. I don't know if you, I'm sorry. Okay. Yeah. So for my people in teams, her question is, how many questions like on the exam are dependent on, like if you know the answer for one question, then that helps you answer the second question. You need to know both. I would say that is probably more in immuno. Like genetics is straightforward. He like teaches you at a really high level, and then he tests you at like a doable level, if that makes sense. For immuno, when you get like bacteria, I mean, you could say it better than me, but for my people in teams, he's saying you see it more in block 2 with bacteria, where you have a question and then the next 2 questions are based off of if you identified the bacteria correctly or like the symptoms or something. So that's more in block 2 and 3 for immuno. But for other classes, I would say that's like not seen that often. Yeah. So your question?
I have a question in regards.
Yeah. So week of exam, her question is, do we still have these practice quizzes the week of exams? Yeah, we did. Yeah, we did. We'll have them for you. We also will write our own practice tests for you guys. So that's great for you guys. I see a question in the chat. Yeah. There's none. Okay, guys, for my people in teams, on Friday, you have your anatomy exam, or I'm sorry, on Thursday, you have your anatomy exam, and then on Friday, we'll do another practice exam like this, but excluding anatomy questions for the remainder of the 3 exams that you have left. And then on immuno, when you take your exam on Friday, there's nothing. Chill, take a break. Okay, so I see a question here. How much time do we get on exams? 70 minutes, right? Yeah. Yeah, okay, cool. Does anyone else have any other questions? Yes.
Hello?
When we make our own practice exams, they are 50 questions like your actual test.
Yeah. Or do you want to? Oh.
I guess it just depends on the TA. So like we would get practice exams sometimes like 2 days before the exam.
Like, are we going to be able to?
So we'll try to be like that week before.
Also, for that first week with the anatomy exam,
1 second. Sorry.
are we just
Yeah, so like that week before. Okay.
where we're
So
not going to be able to have a TA session afterwards?
How do I do that? How do I turn? Sorry, I can't hear you that well. Okay. Oh, wait. Okay. So, anyone on Teams with their question? I don't know who's talking. Hey. I'm still here for you. Okay. Do we still have TA for biochem on the day of the exam? So the TA schedule on exam week is going to change. We will adjust accordingly, and we will send out an announcement with the adjusted TA schedule so that it actually makes sense for you taking your exams. Okay, now I think the person is ready. Kathleen, we can hear you. No, we can't. So sad. Kathleen, do you still have a question? She's leaving. Okay, does anyone on Teams have a question? Somebody has their hand raised. Okay. All right, does anyone in person? Yes. After exam. Mm-hmm. I have no idea. I'm sorry. Yeah. On Teams. Yeah. So for my people on Teams, we're thinking that exam week lectures after exams are going to just be held on Teams because they're not expecting you to drive right after taking your exam, but we're not sure. Double check with Rose. Okay. Will we have an anatomy TA session before our exam to cover the last section? Yes, that will be a part of the adjusted TA schedule. When did you guys all make your study sheets? After the lectures or after the TA sessions that you had? Both, just continuous. Like if I would like take my notes and then just add stuff in based off of the TA sessions or take stuff out based off of the TA sessions. She said exam week TA session. Oh yeah, sorry. Okay, any more questions in here? No? Any more questions on Teams? Okay, guys. Yeah, have a great weekend.
Hi, Ivy.
Oh, hi.
It's Kathleen again.
Oh, yeah, I'm glad I can hear you now.
Yeah, sorry. I was just wondering if we can have